Okay, welcome to Let's Get to the Marks, going through the GCSE Combined Science Trilogy 2024 Paper 2. This is for AQA, educational purposes only. This is to help you get the best level you can in your exam. You'll find all of these on the fantastic AQA website. Also, Physics and Maths Tutor has most of the exam papers, revision packs, notes, and loads of other things to help you. All right, there's 6 questions in this paper. It's an hour and 15 minutes. What I suggest is at the start of each question, you press pause on the video, grab a pen and paper, have a go at the question, and then unpause and watch the walkthrough. Let's go. Question 1. Limpets are small animals with shells. Limpets attached to rocks on seashores. Figure 1 shows limpets on rocks. Here's figure 1. You can see here we've got shells attached to rocks on the seashores. These are the limpets. Limpets eat algae. Limpets are prey for crabs. Give the food chain for algae, crabs, and limpets. So we need to put this in order of a food chain. So what comes first? Well, algae is the producer, so it absorbs the sunlight, does photosynthesis. And then what you're showing is the arrow always goes from what gets eaten into the eater. So the next thing is the limpet, because they tell us that limpets eat algae. And then limpets are prey, meaning they get eaten by crabs. Not crabs, but crabs. There we go. That's going to get you a mark. Hope you did well. Let's move on to the next question. Students estimated the population of limpets on a seashore. The students were given a square quadrat. Here's the quadrat. Figure 2 shows it. Figure 2. We got 50 centimeters that way. Calculate the area of the quadrat in meters squared. Right. Hopefully you know how to calculate area. You're going to do like length times width. So we're going to do 50 centimeters times 50 centimeters. And that would get you the area, but it might be a better idea. I'm going to show you 2 ways to do it. So this would get you 1 mark, and that would give you 2,500 centimeters. Now, and that would be squared. Okay. Now, a lot of people will be like, oh, there's 100 centimeters in a meter. So if I want to change that into meters, what I've got to do is divide by 100. But this is where you'd be wrong. Okay. Remember that a meter square, if we want to change it into meters squared, and this is in meters squared, a full meter square is 100 centimeters times 100 centimeters. Guess how many centimeters that is? If you do 100 times 100 in your calculator, you're going to get 10,000 centimeters squared. So actually, this is only a quarter of a meter squared. We can divide this by 10,000, and that is going to get you 0.25 meters squared. So it's a big difference. If you divided that by 100, you get 2.5 meters squared. How can it be 2.5 meters squared? It's only 50 centimeters long. So it's 0.25 meters squared. Now, another way to do this would be to say, right, I'm going to divide that by 100 and get that as 0.5 meters. Probably the easiest way to do this. And then I'm going to divide that by 500 and get 0.5 and then do 0.5 times zero point 0.5. So if you do that right at the start, you don't have to do the divide by 10,000, and then you get 0.25 meters squared. So I'd say that way is probably easier to convert first. If you convert second, then you have to know that there are 10,000 centimeters squared inside 1 meter squared. Anywho, that'll get you 2 marks. Let's move on. Right, 1.3, the total area of the seashore was 1,800 meters squared. The students sampled 2% of the total area of the seashore. Calculate the number of times the students needed to use the quadrat for the 2% sample. So what we want to do is find out what is the total volume, right? So how much did they want to sample? They only wanted to sample 2% of that. So how do we do 2% of that? When you're timesing by a percentage, you divide by 100, so 2 divided by 100, and then you times the total area. And that is going to give us the volume of the area they actually quadrated. So we're going to go 1,800 multiplied by 2 divided by 100, or 0.02, and that equals 36 meters squared. So that's going to get you your first mark for working out the what 2% of 1,800 is. So it's 36. You can do another way is to say I'll work out 1%. You could divide 1,800 by 100, you get 1.8, and then times it by 2 to get 2%. Some people do that way. That's totally fine. Now what they're saying is how many quadrats did they use? Well, we just worked out that each quadrat was 0.25. So how many 0.25s go in that? Well, I'm going to do 36 divided by 0.25, and that's the same as really saying 36 times 4. So it's 144 quadrats. And it does say use your answer from question 0.12, which is the clue that you had to do something with your 0.25 meters squared you worked out in the previous question. So that's going to get you the second mark. Lovely jubbly. Let's move on. Right, we're on to 1.4. Explain why throwing a quadrat is not a random method to estimate population size. You do not need to refer to safety in your answer. Now, they used to accept shut your eyes and throw randomly as a way of randomly placing quadrats. The only problem with this is students can only throw so far. That like the record for the javelin is only like 100 meters. If you've got a big field, you're not going to throw your quadrat more than about 10 meters. So students can only throw so far. Also, it should be you're trying to avoid bias. So it's got to be random. So you could shut your eyes and spin, but you still might have an idea of where you're letting go, which direction. So it will be biased. They didn't say the student had their eyes closed. Even if they had their eyes closed, they'd still have an idea. Or I'm not throwing that towards my mates. You'd still have an idea roughly where it is. So it would be biased. Right. And let's move on. Describe only one method students could use to plan where the quadrat. Describe one method, sorry, there's more than one method, but the students could use to plan where the quadrat should be randomly placed each time. So what we would say here, you would divide the area into a grid. So you make a little map of the area and number the grid. Now you could say pull numbers out of a hat, pull numbers out of a hat, or use a random number generator. So you basically you take the area, you divide the field up like that, kind of looks like a quadrat itself, and then you number the field, your map of the field like this, and then you can fold up some bits of paper with numbers on, right? Put them in a pocket or put them in a hat, jumble them around, and then you could just stick your hand in and pick one out, open it up, wherever it is, go place the quadrat, or you can just use your calculator to generate a random number. Any of those would be good methods. So personally, I divide the area into a grid, number the grid, and then pull numbers out of the hat, or finish off by saying random number generator. Suggest one hazard the students should, and again, one, we don't want to suggest two. So suggest one hazard the students should be aware of when collecting data on the seashore. Do not refer to throwing quadrats in your answer. So if you're walking around the seashore, what's it going to be like? What are the dangers when you're climbing over the rocks on the seashore? So I'm just going to say rocks will be wet and slippery. You could fall and hurt yourself. Of course, we've also got drowning. If you fall in the water, which is probably the more obvious one, water is incredibly dangerous if you fall in it. Okay, populations of populations of limpets are monitored to assess the impact of pollution in the water. So just one type of pollution in water that may affect the population of limpets. So they're really just asking you for something that a pollution that could be in the water. So we could say oil, crude oil, we could say microplastics, anything sensible. So this is in the news a lot, microplastics. We could say sewage, sewage. Yeah, we talk about these in biology. So sewage, microplastics, crude oil spill, anything like that would go well, really. There's probably tons of these on the mark scheme. So they're chemicals as well. You could say toxic chemicals. I reckon they'll give you toxic chemicals from like a factory, from a waste factory as well. There we go. Right, let's move on. How did you do? That was out of 11 marks. Let's zing on quickly. We're on to question two. Potatoes are a food crop. 2.1, potato plants are classified as eukaryota. Which type of classification group is eukaryota? So eukaryotes, prokaryotes. What have we got? Eukaryotes, and then we have bacteria and archaea. And do you remember what they fall under? So kings play cards on fat girls' stomachs, or king prawn curry or fat greasy sausages. And then Carl Woosey in the 1990s came up with a new level of classification called the domain system. And so our answer here is domain. So the way I remember the order is delicious king prawn curry or fat greasy sausages. You can say millions of other ones. And that came in front, and that split all of the living things up into eukaryotes, bacteria, and archaea, which is what they call them at GCSE. They have slight different names if you go to a higher level. Okay, potato plants can reproduce by asexual reproduction. Which statement is true about asexual reproduction? Meiosis occurs. Offspring are genetically identical. Pollen and egg cells are produced. Right. It's going to be meiosis. I always think of that as E and the S, eggs and sperm. That happens in sexual reproduction, so eggs and sperm. Um, Pollen and eggs are produced. That also you get meiosis with that. So these two are wrong because they're to do with sexual reproduction. And then offspring are genetically identical. Yes, all of the offspring will be genetically identical with potato plants. Okay. When they reproduce asexually. 2.3, flowers of the potato plant contain gametes for sexual reproduction. So what they're saying is they can do both. They can do asexual and sexual. How is a gamete different from other cells in a potato plant? So a gamete is like your egg or your sperm cells. A gamete contains 1/4 the number of chromosomes. A gamete contains half the number of chromosomes. A gamete contains double the number of chromosomes. Hopefully you know this. It's known as the haploid number. So in humans, it's 23. Men's sperm cells contain 23 chromosomes, women's egg cells contain 23 chromosomes, but an adult body cell, like your skin cell, hair cell, heart cell, they contain 46 chromosomes. But your gametes are your sex cells, and in humans, it's 23. God knows what it is in potatoes. I haven't Googled how many chromosomes they have. 2.4, plants in the same genus as potatoes have been studied by scientists. Describe one way a new plant species could be identified as being in the same genus as potatoes. So we've got to say one way that we could tell if a new plant species is in the same genus as potatoes. And what we could do is see if they can reproduce, um, breed them together and see if they reproduce. They should be able to produce hybrid offspring. So that might be one way. Another way is by looking at the DNA. So you can do some like biochemistry where you actually look at the DNA and see how closely matched it is, or you can look at the proteins. We're going to go with breed them together and see if they produce offspring. Now, they might produce fertile offspring. That would mean they're the same species. But if they just produce offspring, they could be hybrid, but that would maybe prove they're definitely in the same genus. Now, actually, thinking about this, I'm not sure if breeding them together will be on the mark scheme. They normally want you to say breed together to produce fertile offspring for the same species. Genus, it's a little bit of a broader group. You could be in the same genus and not produce any offspring. So that isn't a definite answer. I would go for what I was saying before, which is see how closely, or let's say compare the DNA. Compare, uh, compare the DNA. Or we could say compare the structure of um, common proteins. There's a protein called cytochrome C that's found in most living organisms. It's used in part of respiration. So you could compare the structure of the common protein, compare the amino acid sequence. So we could say compare amino acid sequence of similar proteins. God, I can't spell. Anyway, yeah, phylogeny, seeing how closely related things are. That's what the study of this is called. Okay, let's zing on. Let's zing on. 2.5. Scientists have collected and stored seeds from species in the same genus as potatoes. In the future, these seeds may be used for genetic modification of potato plants. Genetically modified potato plants could help supply food to the human population as the climate changes. Explain how the genetic modification of crop plants may be important for the human population to survive climate changes. This is quite a hard question, 6 marks. This is probably the hardest question in the paper because it's a little bit vague. But essentially, what they want you to get at here is during climate change, you don't have to say it in any particular order, but during climate change, the weather, uh, can't spell, the weather, actually, it's my fat fingers. The weather may become colder because the climate might change. The GM potatoes may be able to withstand cold temperatures, or um, if the climate becomes drier or hotter, the GM potatoes could um, have a wider range of climates they can survive in. So what we're saying is it doesn't matter whether the climate gets colder, hotter, wetter, drier. The GM potatoes may have a wide range of alleles or genes that enable them to survive in these different climates. They give them beneficial characteristics, and that's the advantage with genetic modification. Also, you may get new diseases born out of climate change, right? Because it affects which diseases are going to survive. Also, you may get new diseases. The GM potatoes may be more resistant to disease. You could give them a gene that would make them resistant to the diseases you expect to come around because of climate change. We could also say the they may have defenses or chemicals they produce to put off insects. So the potatoes may be resistant to insects or new pests. So maybe there's a chemical you can put in your potatoes, a gene which codes for a protein, which is a poison or a toxin against a toxic substance to insects, but harmless to humans. And you could put that in there. So if new insects come to the area because of climate change, the weather increases, whatever happens that brings in some new pests, the potatoes are more resistant to that. So you could have flooding. The potatoes are able to withstand floods. Um, the potatoes can grow in flooded soil or soil with less nutrients and minerals. I should really just say minerals. Let's not say nutrients. That's not really how plants get their nutrients. They make their own nutrients, but they absorb minerals through the soil. So, what else? So we're just basically sort of saying all the changes that happen in climate change, the hot weather, the cold weather, the floods, all of this, the potatoes, um, could be important. Also, the GM potatoes, um, would be important to feed a growing population. Um, they might require less space. So they would require less space, right? What are they doing with these potatoes? So they've got the seeds stored. So again, we want to say storing seeds uh, takes up less space than storing potatoes. They'll also last longer. They won't rot. It's easier to store seeds. So we could also say easier to store seeds um, than plants. Potato plants. Right. So if you were trying to keep potato plants alive in an area, it's much easier to store seeds. They just need to be kept dry, away from water and cold or cool, and then they won't germinate. And you could store millions of seeds, whereas millions of potato plants would require watering every day and a lot more effort. So actually, storage of the seeds is a really important thing. And the fact that they're GM means they can withstand different changes in the weather and different pests and different diseases. And that should be enough. But yeah, it's a difficult question, this one, because it was quite vague. Um, you had to have quite a good understanding of conservation. Okay, question three. AKU is a genetic disorder. Figure three shows the inheritance of AKU in one family. So we've got A and B here. And then we've got C, D, E, F, and G, H, I, J, K, L, and M. The dark square is a male who has AKU, which is this genetic disorder. The dark circle is a female who has AKU. The light square is a man who does not have AKU, and the white circle is a female who does not have AKU. Describe how figure three shows that the allele for AKU is recessive. Well, let's check out. We can pick on any, when you want to find out if you're dealing with a recessive or dominant allele, look for two parents who don't have the disease, i.e. A and B, and then check out one of their kids has the disease. That tells you that it must be recessive. We've got the same thing going on here with F and G and either L or K. So you're going to talk about that or you're going to talk about that. And so what we can say here is A and B do not have the disease, but they have a child D who has the disease. This means A and B, I need to just open this text box up. Sorry, guys. This means A and B are carriers. Okay, so if we were to assign, just to explain that to you, if we were to assign A and B some letters, think about it. Let's pretend it was dominant. So let's pretend it was dominant and found on the big T. Then if they had a big T, they would be they would have the disease, so they'd be colored in. But they don't, they're not colored in, so they don't have the disease, and so it can't be a dominant disease. And the reason is um it must be found on the little t. So they have to be big T, little t, because if the little t carries the disease or the allele, the recessive allele gives you AKU, then these two are totally fine because they have the big dominant normal allele. But it shows that they can have a child that has the disease, right? If it was dominant, then they would have the disease. You can't have a dominant disease in which someone is carrying one of the alleles and doesn't have the disease, essentially. So A and B do not have the disease, but they have a child D who has the disease. This means A and B are carriers. So that would be they're carriers, people who have the allele but not the disease, and that can only be with recessive conditions. Okay, let's move on. Let's move on. Which personally is definitely heterozygous for AKU? Well, we know A and B must be heterozygous for AKU, but let's check out C, D, E, and G. Well, again, we can't tell with C. We can't tell. Well, we know D, right? They gave D as an option, right? So they've gone C, D, E, and F. Let me go along and explain it to you. You might want to pause the video, have a go, guess which one is heterozygous. Now I'll explain it. Okay, well, H and I don't have the disease. We know that D must be little t, because we said it's a recessive disease. I've just picked T, by the way. I probably should have done A as it's called AKU. You might be getting a bit confused by that, but it doesn't really matter, whatever capital letter. So this person doesn't have the disease. So we know they have to either be that or that. So they could be big T, big T. Their children don't have it. So we can't conclude which one they are. E, we've got no idea what E is. If this is big T, little t, big T, little t, they could be big T, and big T, little t. So we have no idea what E are. They could be big T, or big T, little t. You might think the same with F, but look, F has had K and L. That means two recessive alleles have come together to form K and to form L. So as they don't have the disease, we know F must be big T, little t. And in fact, so must G be big T, little t. But it's asking us F, I think, was one of the options. Let's go and have a look. So they are definitely heterozygous. Oh no, they're asking us about G, but it's the same thing. So G had to be big T, little t because their kids had the disease and they didn't. Right. Zing. 3.3. A female has AKU and a male who is heterozygous for AKU plan to have a child. Determine the probability the child will have AKU. You should complete figure 4. Identify the phenotype of each of the offspring's genotype. Use the symbols right. We're not going with big T, little t, which was done by me. We're going with big A, little a. So let's do that. Right. So the female has AKU and the male is heterozygous. Now we've already worked out that then she must be, it's a recessive disorder. So she's little a, and the male is heterozygous. So he's big A, little a. We want to know the probability. Now I'm going to show you the quick way. You basically, you just pair them up like that. Big A, little a, little a, little a, little a, little a. Now you're going to be tempted here to say, oh, it's 50%. And you'd be right. It is 50% because these ones have the disease. But we need to let the examiner know that when you say, oh, it's 50%, or you say, oh, it's 0.5, oh, it's 2 in 4, um, we need to tell the examiner, or we need to tell the examiner we know which two are the ones with the disease. Because the examiner might think that you think big A, little a is the person with the disease. So that extra mark is for you identifying this person has AKU. And this person is just a carrier. I don't probably just have to identify the AKU person. So this is a carrier. This person has AKU. So there's a 0.5 probability that they will have AKU. Now, what do you get the marks for? You get the marks for working out mum and dad's genotypes. You get the marks for working out the genotypes of the offspring. And then you get the marks for the phenotype, the carrier and the AKU. And then you're going to get the marks for the probability. And there's your 5 fat marks. Boom. Right, 3.4. A mutation is a change in a gene. People who have AKU have a mutation that causes the production of a non-functioning enzyme. Enzymes are proteins, suggest how a mutation can result in the production of a non-functioning enzyme. So what is a mutation? It's a change in a gene, and this means it codes for different amino acids. So you get a different protein shape. The active site of the enzyme no longer fits with the substrate. So the shape of a protein is decided by the DNA, the genetic code, because remember, genes code for an amino acid sequence, and they give us the specific, the amino acid sequence helps form the specific 3D shape of a protein. If you get the sequence changed slightly, the shape of the protein might be changed, and it'll be the active site of the enzyme that no longer fits with the substrate, and so it's now non-functioning, that enzyme. Right, let's zing on. Sorry about my sniffles. 3.5. Some people have in vitro fertilization, known as IVF, treatment to increase the chance of becoming pregnant. Describe how the process of IVF can result in a pregnancy. So we've basically just got to describe IVF. So female has hormone treatment. So what hormones does she get? She gets FSH and LH to increase um, maturity. You can say ripening, developing of eggs, and release of eggs. Eggs are removed in a procedure and mixed with man's sperm. Okay. Once embryos have formed, the best are selected, and should only be one implanted. They sometimes implant more than one, but really most of the health services now only implant one embryo because of the risks of multiple births with more than one embryo, which gives more risk to the mother and the children. Once embryos have formed, the best are selected, and one is implanted into the woman's uterus. So it's all about the keywords. Now, what I didn't say is fertilization, which was a big boo-boo by me. So eggs are removed in procedure and mixed with the man's sperm. So fertilization can occur. There we go. We just want to get those keywords in. Biology is a keyword game. So I've explained the hormone treatment. I've then said mix the eggs and sperm, fertilization. They form an embryo and then implant them in the woman's uterus. Boom. Bish bash bosh. Let's move on. Another sniff. Call me the sniffer. 3.6 embryos no, don't call me the sniffer, right? 3.6 embryos can be screened to detect inherited disorders. Give two arguments against embryo screening. Do not refer to religion in your answer. So there is a risk of miscarriage. So there's a small risk. So the actual screening to check if the embryo has any genetic disorders could actually cause the baby a miscarriage to occur and you lose the baby. So that would be devastating. It's a very small chance, but it's going to happen every year to some people. They're going to have a miscarriage from the embryo screening because although the chance is small, there's lots of pregnant people. So every year that is happening. But when you weigh it against the risk of not knowing if your child has a genetic disorder, most people make the decision to have embryo screening. What's the other risk? The couple, the parents, may not keep the baby, or the screening might be wrong. It may cause distress to the parents. So there might be all sorts of things which, like imagine if it's actually wrong and you have an abortion and actually, and then of course there is a moral argument, although there is a moral argument, the embryo does not have a choice. But some people would say that's dumb because it doesn't have a neural network, i.e. a brain, so it can't think. So, but people are saying, well, look, you're taking the choice that if you gave that future human a choice, like you've been born with this disability or this genetic disorder that may limit your life, but would you still want to be born? It might say, yeah, hell yeah, I want a chance at life. So we're not giving it the choice. But in a way, you could also argue, well, it doesn't have a brain or a nervous system yet. So it's a silly argument, but that is an argument that people put forward. So um, there we go. Right, let's move on. Question 4, rat populations can increase rapidly. When rat populations are large, there is competition between rats. When rats compete, the basal metabolic rate of the rats increases, right? So when they're competing with each other, their metabolic rate goes up. Basal metabolic rate is controlled in the same way in humans and in rats. Describe how basal metabolic rate is increased. So we might go, oh, so basically, you just need to say, how does the metabolic rate increase? Now, there's more than one way to increase your metabolism, and there are two that are mentioned in the AQA syllabus. So we've got thyroxine hormone that you get the pituitary gland releases TSH, thyroid stimulating hormone, that goes to the thyroid and stimulates thyroxine hormone is released from the thyroid gland. So that is one reason. And then another, that would get you 2 marks. And then another one is adrenaline is released from the adrenal gland. So both of these increase the basic or basal metabolic rate. So that'd get you 2 marks, that'd get you 2 marks. And both of these appear in the AQA syllabus at different points. Okay, one size of rat population, the size of the rat population can increase quickly. One female rat, one male rat can produce 20 offspring every 2 months. Warfarin is a poison that's been used to control rat populations. Rat populations can become resistant to warfarin by the process of evolution. A population of rats was given warfarin for 4 months. Table 1 shows the information about the resistance to warfarin in the rat population for a year after the warfarin was first given. So, number of months since warfarin was given. Was warfarin being given? Yes. Percentage of the rat population resistant, 1%. Then after 2 months, 4 months, look, after 4 months, 81% of the rats are resistant to warfarin. And then we can see they haven't been given warfarin for ages. And what is happening? You take away that selection pressure and the percentage of the rats resistant is slowly going down. It's no longer a benefit and no longer makes you more likely to survive. Rats with resistant to warfarin have a similar mean mass than rats that are not resistant to warfarin. So their mean mass, oh no, sorry, rats with resistance to warfarin have a smaller mean mass than rats that are not resistant to warfarin. Smaller rats are often at a disadvantage when competing against larger rats. Explain the trends in the data when the warfarin was being given and when the warfarin was no longer given. So this is a bit of a natural selection question. So it kind of goes like this. When how do you spell that bad boy? When warfarin is present, the smaller rats, think about natural selection, have a greater chance to survive. Remember the natural selection answer? Breed, pass on the small rat allele, just made that up, but the allele or the gene for being a small rat to their offspring. This happens over many generations. So we see an increase in small rats resistant to warfarin over the first 4 months when warfarin is present. So while the rats are living in an environment with the warfarin, which basically kills the rats, the smaller rats have a greater chance to survive because they're better. They're more resistant to warfarin. Pass on the small rat allele. I should say pass on the resistant allele. Pass on the small rat. Let's call it the resistant allele to their offspring. This happens over many generations. So I would say there's at least 3 or 4 marks. Now we need to explain when the warfarin is taken away, when the warfarin is taken away, we can see a decrease in the population or in the number of small rats or warfarin resistant rats. This is because now it is an advantage to be bigger. And the bigger, less warfarin resistant rats are more likely to survive, breed, and pass on their genes to offspring. So it's really asking you about natural selection, but to explain, look, generally, when there's no warfarin knocking about, the advantage is to be a big bad boy rat. The big bad boy rats, they can fight better, outcompete the other rats, steal more food. They're like, yeah, I'm gonna survive, you're gonna starve. They're like bigger, badder, right? But if you throw warfarin into the mix, suddenly being small is an advantage because the smaller rats tend to have a resistant gene towards warfarin. And so if you throw warfarin in, they're like, oh, you big boys are in trouble now. And they're more likely to survive, breed, have sex, have children, pass that small rat resistant warfarin allele to their children. And so they increase in the population, but take the warfarin away again, and we shift back and they start decreasing in the population. And the bigger rats that are less resistant to warfarin start increasing because we've got new selection pressures like fighting for food and stuff like that. Whereas when you introduce warfarin, that is the biggest selection pressure. That is the biggest thing that is basically allowing them to survive. All right, that was a difficult question. The six markers in this 2024 paper are hard. I have to say that. Right, question five. Otters are mammals that live in river ecosystems. Figure five shows an otter. Define the term ecosystem. So an ecosystem is both the abiotic and biotic factors in a habitat or an area. So an area, all of the abiotic and biotic factors in an area or habitat. Oh my God, I'm still sniffing. 5.2. Otters are important species for the stability of the river community. Describe two ways the animal species may be important for the stability of the whole community. So, what is it that the otters, why might they be important for the stability of the whole community? It just shows an otter. So, this is a difficult question. You have to know your conservation stuff. So, the main ones are that otters may be prey to a predator. I don't know what eats otters in the UK, whether there's foxes that eat them or whatever, but otters may be a prey to a predator, so a food source, but also otters may be a predator themselves, right? What do they eat? Fish? I'm not even sure. Otters may be a predator that keeps, whoopsie, a prey population under control. So that's important because if their prey gets too big, it may damage the ecosystem. Otters also may alter the environment. Now, I get mixed up with otters and beavers for some stupid reason, but I can't remember what otters do. But they might make their dens or wherever they live out of sticks, and they may provide habitats for insects and other things. So also may alter the environment to provide habitats for other species. My knowledge of zoology is poor. But yeah, their waste may, the otter's waste when they go for a poo, basically their feces may provide minerals for plant growth. So there's all sorts of ways that they could influence or, sorry, support the stability of the river community. Sewage was accidentally added to the river. The sewage moved with the river and affected the number of bacteria in the water. So sewage affects bacteria in the water, the concentration of oxygen in the water. Samples of the river were analyzed at different distances from the sewage. So we got 0 meters from the sewage, we got 4 times 1,000 bacteria or whatever that is per millimeter, oxygen concentration 5.4. And then as we get further from the sewage, the number of bacteria is increased. So we've got 216,000 per millimeter cubed. And the oxygen concentration has gone down. So there's a link there between the number of bacteria and the oxygen concentration and the distance from where the sewage was added. The number of bacteria at 5 kilometers was greater than the number of bacteria where the sewage was added. Calculate how many times greater. So the sewage was added here and was 4. And then at 5 kilometers, it's 216. So we just want to do 216 divided by 4. Wasn't that Stone Cold Steve Austin? No, maybe not. 216. it was something else. Wrestling fans. Okay. A student concluded the number of bacteria is proportional to the concentration of dissolved oxygen. Explain why the student's conclusion is not correct. Use table two. So we have to use the table. Now, this symbol means proportional. Let's see, we can prove that this is not true because we can say you can pick any two numbers and prove it's probably not true, right? So um, if I go da, 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 5.4 divided by that, you should always get the same. Yeah, you can show it's not true. You can show it's not true. So how are we going to show it's not proportional? So when the bacteria was 4,000, that was 5.8. When it was 75,000, that was 4.8. So yeah, what we want to do is put that into fumbling around for the words here, but essentially, when we double, the way to look at this, we got to, is there a doubling at all? Oh my God, it's not set out very nice, is it? It'd be lovely if they had a doubling, but they don't. But essentially, it's not proportional. It's not in the same ratio. So like when I double the number of bacteria, or we haven't doubled it, but whatever we've done there, so 75 divided by 4, whatever that is, 15, 16, 17. Well, Sugar Ray Leonard. Let's do 125 divided by 4. So if I look at this, 125 divided by 4. So if I go 125 divided by 4, that has increased by 31.25 times. So it's got 31.25 times bigger. Has this got 31.25 times smaller? So as that has gone, has that gone 31.25 times smaller? No, it hasn't. It's gone about 25% smaller. It's not even gone anywhere near. That's gone like way bigger, and that's only a little smaller. So we can say they have an inverse relationship. So as the number of bacteria increases, the amount of oxygen goes down, but it is not in a proportional ratio. So they're not in the same ratio, is what we want to say. God, I made a meal out of that. So as the number of bacteria increase, the oxygen concentration can't spell goes down. Don't worry about that because we're all about speed here. However, it is not in the same ratio. It's not in the same ratio. We'll go with that. It's not proportional. Okay. Or you could say there's a negative correlation, but it's not in the same ratio. 5.5, otters live in water and on land, eat mainly fish. The concentration of dissolved oxygen has decreased in a river where the otters live. Explain how the decrease in concentration of dissolved oxygen in the river water will affect the population of otters. So let's think about if the oxygen concentration goes down in the river water, what's going to suffer? What lives purely in the water? And you should come up with the answer fish. So fish have less oxygen. What for? What is oxygen needed for? For respiration. I could say aerobic respiration. I'm just going to put for respiration. So fish have less oxygen for respiration. More fish die. Less fish for otters to eat. So otter population decreases. So the main thing here is fish have got less oxygen, so fish population goes down, less fish for the otters to eat, so their population goes down. Bosh. Right, we're on to question 6. Hormones are released from endocrine glands. Which gland releases the hormones to control other glands? Which one is known as the master gland? It's the pituitary. So it controls this with thyroid stimulating hormone, and it controls the thyroid with TSH. That's the example you need to know. Several hormones can affect the blood glucose concentration. Adrenaline can increase the blood glucose concentration. What is one other effect of adrenaline? So, when your adrenaline is released into your blood, does it make you breathe slower and calmer? Does it slow down your metabolic rate? Does it increase the amount of eggs getting ready, um, maturing more eggs or developing more eggs, or increase blood flow to the muscles? It's known as the fight or flight hormone. So if you were going to have a fight or run for your life, would you want slower breathing? Would you want slower release of glucose? Or would you want more blood in your muscles? Well, you're going to want more blood in your muscles because it gives you more oxygen, more ability to contract those muscles and run for your life or fight for your life. So that's how we work it out. Would you need it in a fight? If you do, adrenaline's your man or your woman. 6.3, cells in the pancreas detect changes in the blood glucose concentration. What type of cell in the body detects changes? Think about the nervous system. You'll mainly think about this cell in the nervous system. What detects a stimuli? So which type of cell detects a stimuli? It begins with the letter R. It's a receptor cell. Okay, now I know this wasn't about the nervous system, but we have receptor cells. They receive information and they detect changes in the environment. Scientists investigated whether blood glucose concentration affects reaction time. The reaction times of 10 people, is that a lot? With type 1 diabetes were measured. This is the method used. Tell one person to drink glucose, monitor the person's blood glucose concentration, record the person's reaction time when the person's blood glucose concentration is 16, 6, and 3. Repeat steps 1 to 3 for the other 9 people. Okay, but did they do 3 repeats at each concentration and take an average? Who knows? People with type 1 diabetes were selected for the investigation instead of people who did not have diabetes. Why? This is because people with type 1 diabetes, what don't they produce? Um, they cannot produce um, insulin. Um, so the glucose concentration will remain at the level being investigated. So if you gave someone a 16, their blood glucose concentration is going to stay at that level for longer because they don't have insulin. A normal person will have insulin. A normal healthy person with a healthy pancreas will have insulin that removes this sugar and puts it into the liver and the muscles. So the blood glucose concentration will not stay at that level for very long, a few minutes at most. Whereas somebody with type 1 diabetes, their blood sugar concentration is going to stay at that level. Whether it's ethical to be giving diabetic people sugar drinks is another matter. Right. Um, let's move on. Control variables between the different people in the investigation. So they controlled the age and the sex of the people, and the food and drink before the test suggest 2 other variables that should be used in the investigation. So what 2 other factors? They controlled what food they ate and what they drank. They controlled whether they were a male or female. They controlled their age. What other factors might be linked with diabetes? So let's say body mass, because we know that obesity is linked with diabetes and things like that. So I would definitely put body mass there as one of them. What else should we do? Now, we have to remember that these people are doing a reaction time test. So, what sort of things would affect your reaction time? Obviously, they've said food and drink. So, I suppose we can't really say caffeine, maybe, but tiredness affects reaction time. Um, Distractions, are they listening to music? That kind of thing. Genetics, genetics or family history. Um, uh, yeah, we'll go with that. We'll go with that. Whatever, whatever. I don't think drugs include in the food and drink caffeine, but who knows? It was important for scientists to monitor the health of each person during the investigation. Well, they're diabetic, so they could slip into a diabetic coma. They could get sick as diabetic, could have a diabetic coma. That's a terrible answer, like the way I've worded that, but you know what I mean. They can't control, they do not produce insulin, so they high sugar levels is dangerous. High blood sugar concentration is dangerous. So, well, I said diabetic coma, so that's good enough, I think. But yeah, it's dangerous because you could go into a diabetic coma. So let's sing on to the last question. Here we got, no, it's not the last question. Here we've got figure six shows the results. We've got the reaction time in milliseconds, and we've got the blood glucose concentration. So, okay. People who do not have diabetes usually have a blood glucose concentration in the range of 4 millimole per decimeter to 7.8 millimole per decimeter. Okay, so they're normally 4 somewhere there to 7.8 somewhere there. Describe how the results in figure 6 show the importance of homeostasis. Um, So this one's quite hard to interpret this, but basically what they're saying is that the reaction times here seem to be lower with people. Look at the reaction time. So this reaction time is quicker, i.e. less time. They react faster within the normal blood glucose concentration range, which is like that. People either who have too low sugar or too high sugar, they don't react as well as people with the normal blood sugar concentration. So homeostasis keeps the blood glucose concentration within a narrow range. So it keeps the blood glucose concentration within a narrow range. Um, normal I shouldn't really say normal people. People with diabetes aren't normal. People within this range, I should just say, people within this range um, have quicker reactions. Why is it a good thing to have quicker reactions? So more likely to survive. If you think about natural selection, more likely to survive, breed, pass on genes to their offspring, all of that business. But they're more likely to survive, less likely to get damaged by something in the environment. Right. Let's zing on. 6.8, describe how blood glucose concentration is maintained within the narrow limits in people who do not have diabetes. So, yeah, we're going to do that in a sec. What? Oh, okay, let's go. So, oh my good gosh, we put this into words. So here is your pancreas. Essentially, when your blood sugar goes high, so when blood sugar goes high, like after a meal, oh my God, I'm making this such a long thing. So, after a meal, what happens? Your blood sugar goes high. Let's just call it sugar. It should be glucose, but let's go. Blood sugar is high after a meal. What does the pancreas do? The pancreas releases insulin. Now, I think of the IN as in and then liver or liver in, right? So it's going to take the sugar and stick it in your liver, okay? And it turns it into something called glycogen in the liver. Now, after exercise, so after you've been training, um, or after not eating for a while, your blood sugar is low. Hopefully, you know a different hormone is released from the pancreas, and it begins with a G, and it's called glucagon. Glucagon. And I think of that as saying glucose gone. And what it does is it makes glucose gone from your liver and puts it back into the blood, raising your blood sugar back to the normal level. Anyway, that was some crusty ass diagram. Let's just put that down into words. So we want to say after a meal, um, blood glucose level is high. Better to use the word concentration. But there we go. I've said level. Let's leave it at that now. Insulin is released, whoa, baby, from the pancreas. Okay. Insulin puts the glucose in the liver where it is converted into glycogen. Now, do you have to put all of this to get the 5 marks? Probably not, but I want to cover it all, right? So my key points here are blood. When the blood glucose is high, insulin, 1 mark, is released from the pancreas. The glucose goes into the liver where it's turned into glycogen. You could say the liver and muscles, folks, before people go, and the muscles. Right. I'm going to just say after exercise or when blood glucose concentration is low, glucagon released from pancreas. Glucagon um, activates, I mean, it's a bit fancy word really for GCSE, but the glucagon activates the liver to convert or turn glycogen into glucose, and glucose released from liver. Oh, look, I'm going to put muscles in as well, and muscles, raising blood glucose to normal level. Again, it should be blood glucose concentration, but I don't want to be typing from here to eternity. So again, if I was marking this, you're looking at when the blood glucagon is released from the pancreas. Glucagon activates the liver to convert glycogen into glucose. Glucose is released from the liver and the muscles, and the glucose level goes back to normal. So that's what I'm looking for. I'm looking for at least 5 ticks there, and I want to get at least 3 on one and 2 on the other. So, like you want to kind of talk about both situations to get all 5 marks. So you might have scored like 3 or even 4 marks talking about the release of insulin, but you've got to at least mention glucagon and what it does, even if you don't go into the full depth to get the 5 marks. 6.9, the scientists gave the hypothesis optimum blood glucose concentration increases the production of chemicals that diffuse across the synapses. Describe how an increase in chemicals could decrease reaction time. So, optimum blood glucose increases production of the chemicals that diffuse across the synapse. Describe. So, more neurotransmitter. We can just call these chemicals. Um, it actually tells us that increases the production of chemicals that diffuse across the synapse. Um, across the synapse um, between neurons. So, faster formation of electrical impulse. Um, electrical impulse gets to effector more quickly. Yeah. I think actually, I'm going to delete all of that. When I think about it, the production of chemicals that diffuse across the synapse, the key word there, I think, is diffuse. So if we have more chemicals here that are going to diffuse across the synapse, we're going to say there is a steeper concentration gradient. The chemicals will diffuse faster. So more chemicals or neurotransmitter means a steeper concentration gradient. So the I'm going to call it neurotransmitter just to be fancy, but it's the same as just saying the chemicals. So the neurotransmitter diffuses faster across the synapse. And then on the other side of the synapse, it reforms an electrical impulse, and we'll just say the electrical impulse um, arrives at effectors more quickly, just to cover all the bases, right? So there we go. That's the end of the test. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.